Hello everyone, my name is Zahra Mazidi. I'm in this in3 project ESR number 3, and as you can see here, I'm coming from Iran and now I'm in Vienna and doing my PhD in Evercyte company under supervision of Johannes and Regina Grillari. Just to talk a bit about myself and introduce myself a bit more, I did my undergraduate study in molecular and cellular biology in Khwarezmi University in Tehran, and also my master's studies is in developmental biology, was done in Royal Institute of Stem Cell and Technology, again in Tehran. And my master thesis was about disease modeling of systemic sclerosis disease, and we were trying to induce iPS cells from patients-derived fibroblast cells, and then we differentiated them to endothelial cells as well. So here today I want to talk to you about mostly two of my PhD studies that I was doing it through my PhD. The first study is the effect of cyclosporin A on functionality of endothelial cells differentiated from iPS cells as in vitro toxicology model system. As you can see here, this study was a collaboration between in vitro and in silico parts of in3 project. And I'm pretty sure that until this time that you're hearing and seeing my presentation, you already got familiar with in3 project, in vitro and in silico integration. So as you can see here again, I'm doing this project with my other colleagues, Nicoletta, Zahra, Cormac, Christian, and Liadis. And we are doing it in two parts, in vitro and in silico. So as I talk to you about it, and you can see here, the aim of the study is to understand the toxicity of cyclosporin A on endothelial cells. But we were also trying to get close to develop an AOP about it. So I'm not telling that we now have a quantitative AOP or we could model our AOP, but we tried our best to get close to this concept. And that's where our in silico people are helping us. And we can talk about it later on as well. And we can just say something that, for example, Christian developed a tool that two of our key events are in this tool, which helps us to develop more our AOP in a very wide variety as well. So to be able to do this study, at first point, we wanted to have the cells, endothelial cells. So for that, we developed a protocol to induce, to differentiate endothelial cells from iPS cells or pluripotent stem cells. And this protocol, as you can see here, is a short time protocol. So it just takes about 8 days to have the mature endothelial cells. And this is a protocol based on signaling pathways. So we try to mimic the developmental biology of the cells when they are developed in the body, in human body. So in first step, we treat the cells with shear to activate the Wnt signaling pathway to have the mesoderm lineage at day 1. And then we treated cells with some growth factors and small molecules to derive at day 4 endothelial progenitor cells, and then we differentiated them more to have at day 6 late endothelial progenitor cells, which are characterized by VEGFR2 or KDR marker, and then we sorted these KDR positive cells, which were about 30% of the population, and then we treated them in adherent culture, because the first 6 days were in aggregates in collagen coated plates and in EGM2 with media, which is an endothelial specific media, for 2 more days, and then we had endothelial cells. As you can see here, we could characterize these endothelial cells. Here you see a flow cytometry of CD31, which shows that the population of our cells are homogeneous and they have CD31 expression. And here in right down part, you see the iPS cell aggregates at day 0, and then endothelial clone-forming cells. This is the other name of KDR positive cells or VEGF receptor 2 positive cells. And then we have mature endothelial cells, which are the cells that we characterize them by CD31. Also, we did a VWF expression for them to see that they are positive for endothelial cell markers, and we also measured and saw CD31 expression in immunofluorescence studies. As you can see in left part, we have a matrigel assay, and we did a sprouting assay for them to show that these endothelial cells are also functional. So matrigel assay is showing that endothelial cells start to migrate and form some tubes in the plate, and a sprouting assay is a semi-3D assay which was developed in Evercyte when I started my work there, and it shows that cells, when if they are functional and if we treat them with angiogenic factor, that here for example we used VEGF, we see that these cells are having some sprouts coming out from the spheroids of the endothelial cells, which is the first step of angiogenesis. And the good thing is that it's somehow a semi-3D assay. So after having these cells, endothelial cells in culture, and we could passage them and we could keep them until 10 passages. So we wanted to start our study with cyclosporin A. So for that, we tried to develop a putative AOP with the help of our colleagues. So, the putative AOP is, as you all know, is containing a molecular initiating event, key events at cellular level, tissue level, and also adverse outcome. Our molecular initiating event would be binding to cyclophilin D, and then after that, cyclosporin A is binding, or any chemical, because we don't develop the AOP based on the chemical. So after binding, then they inhibit calcineurin activity, and then we should see some mitochondrial dysfunction, and this mitochondrial dysfunction might induce ROS activation, which causes the oxidative stress response pathway on endothelial cells. And then we have two steps in two different pathways. In one pathway, we have the reduced production of VEGF, and then we see an angiogenesis impairment. And in the other way, we see a toll-like receptor 4 activation signal and NF-kappa beta activation signal as well. And then we get again to angiogenesis impairment. So about the MIE binding to cyclophilin D, as I can show you also at the end of the study, we have a strong essentiality, and we have a strong literature review about it. In this study, we are focusing on it, so don't assume that we did not do anything about it. But the thing is that for now, we did some Western blot to detect it in the cells, but the data is not that ready yet, so maybe we don't hear a lot about it for now. But my colleague Nicoletta also did an integration analysis of this putative AOP, and she tried to find a link between this adverse outcome pathway for angiogenesis impairment to existing qualitative AOP in AOP network, and she searched in AOP wiki for that. And as you can see here, in left part we have a developmental deficiency, and these are these two AOP about it, which comes to VEGF, and this comes to, for example, reduced dimerizations. And in the right part, we see an immunoresponse deficiency, and as we can see here, we were lucky to be able to link our AOP into these two available AOP in AOP wiki. So after having them, we started to do our analysis with cyclosporin A. At first, we wanted to see if we can track cyclosporin A in endothelial cells. And so we treated the cells for 3 hours with our cyclosporin A as we got it from literature reviews, because we thought that it was shown in literature reviews that after 3 hours of cyclosporin A in vivo, we see some effects. So what we did is that we measured about overnight about 11 cells, so 11 cells control, which are just endothelial cells without cyclosporin, and then with cyclosporin A treatment. And as we can see here, for example, if we look at the PCA, we can see that all these cyclosporin A treated cells could be clustered together in one plot in one place. And about the controls, we are improving it by doing some more replicates about it. But as we can see here, we see that some of the controls are very well clustered together and 4 more controls are clustered together, but somehow they are still could be clustered together. So we are improving it. So here in the left bottom part, you see that we can see a peak of cyclus point A, and we can see where cyclus point A is. We can track it with Raman microscopy, and here in the right upper part we see a cyclus point A accumulation in treated cells in around the nucleus of endothelial cells, and here are untreated cells. So then we started to go to see the cytotoxicity of the cyclus point A on endothelial cells. So the left part shows the cytotoxicity, which is the resazurin assay. And we did it for 24 hours, 48 hours, and 72 hours. And as you can see here, this differentially expressed, so these iPS-derived endothelial cells, we call it ESR3 because the source, the iPS cells that we drive these endothelial cells from are called SPAD03 colon. And these HUVEC-2 cells are the HUVEC cells, HUVEC endothelial cells, which are immortalized in Evercyte, and we use them somehow as to have a control and to have another cell line to see if this cytotoxicity can be robust across the cell lines. And as you can see here, in both cytotoxicity data, we see an IC50 of around 5 micromolar after 72 hours of cyclus point A treatment. So then we wanted to see if the cyclus point A treatment with this concentration and time point can already affect our mitochondria of endothelial cells or not. So we did this study in Ludwig Boltzmann Institute in Vienna, Austria, that I have to thank my colleagues because of their very nice collaboration in this study with us. So as we can see here, at first in right part, upper part, you see that it shows the oxygen uptake by mitochondria after 72 hours with 5 micromolar of cyclus point A. And as we can see here, the oxygen uptake is significantly decreased, but we knew that at this time point and this concentration, we already missed 50% of the cells. So we thought maybe this increase is somehow although we know that this is more than half, but still maybe the dead cells are giving some signals that it's affecting the other cells for mitochondria activation and oxygen uptake. So we decreased our time points to 24 hours. And as we can see here, we check 2 concentrations of cyclus point A, which are 2.5 and 5 micromolar. And in both of the cell lines, we see an OXPHOS increase by putting these concentrations of cyclus point A for 24 hours on endothelial cells, which means that OXPHOS is increased on endothelial cells. And if OXPHOS is increased, so it seems that mitochondrial transition pores are blocked or somehow affected, and that's why OXPHOS is increased in endothelial cells mitochondria. And we knew if it is the case, so ATP should be synthesized in endothelial cells now after putting these concentrations. So we did an ATP assay, ATP synthesis assay, and as you can see, in low concentration, even in very low concentrations of cyclus point A, which is the 0.6, 1.2, 2.5, and 5 micromolar of cyclus point A, we see an increase in ATP synthesis, which was cool for us because it was coming parallel to our hypothesis. And then, again, thanks to my colleagues at Ludwig Boltzmann Institute, we wanted to see, okay, if it's the case and mitochondrial transition pores are closed, OXPHOS is increased, so we have to see an increase of ROS in the cells and in the mitochondria. So at first what we did, the data that I'm showing you, it's now just for endothelial